have our second presenter here with us today. Okay. He is Professor David Adetitun, a distinguished Fulbright scholar and an Association of African Universities, AAU scholar. He's also a dedicated academic who lectures and supervises students at undergraduate, master's level, and doctoral level, fostering development of future researchers and professionals. He also leads an active research group comprising of graduate students based in Nigeria and India, and collaborates with researchers across various campuses, both within and outside Nigeria. His research focuses on environmental microbiology, addressing critical issues related to microbial interactions and their impact on the environment. Please join me in welcoming Dr. Adetitun for his presentation.
Yeah, thank you, Blessing. Good morning, everyone. Because of the internet disruption, I would like to off my camera as I go on with the presentation. I want to welcome.
I will share your the screen for you.
All right, thank you. Yeah, thank you, Professor Malcolm, for that presentation on qualitative data collection. I will be speaking about quantitative data collection methods, understanding structured numerical data for research. In the course of my speaking, I'll be talking to us and making us understand the concept and characteristics of quantitative research. We will also be able to identify and differentiate types of quantitative data. Then we will be able to learn various methods for collecting quantitative data. Also, by the time I'm done, you should be able to explore how to design effective instruments and sample techniques. Then I will also talk about ethical considerations in data collection. I will take a look at the difference between quantitative research and qualitative. Number one difference is in terms of the objective. Quantitative research deals with quantity, tests, hypotheses, and it generalizes. I'm on the third slide now. I'm on the slide with the blue and white. Yeah. Then qualitative research, in quantitative research, you gain deeper understanding and then you explore context. The quantitative research is systematic and is structured, while qualitative is flexible and open-ended. Then quantitative research uses statistical analysis, but qualitative uses thematic and content analysis. All this we are going to look at as we go on. Then quantitative research emphasizes objectivity, while the qualitative acknowledges subjectivity. Before I go on, as Professor Marcus has said, you can do better work than others, and then you need to be truthful. Most times, students are working or researchers are working, and they want the work to be just the way others have reported it, just the values others have obtained, just to go in line. Whereas in your own research, you may be discovering a new thing, and we have had cases like that in the past where our results were different from those of others. What I'm saying is that just be truthful with your work, and then the rest is going to take care of itself. Your results may or may not be different from those of others. Then Professor Malcolm said this also, that you should share your results with those who provided data for you. You've gone out to get data from certain individuals, and then they allowed you to get the data, maybe from their establishment, maybe from their any kind of maybe animal or plant, or even themselves, or you sample the environment. By the time you are done, you should go back and give them feedback. You know, I will supervise some master students in air microbiology, and then the students were asking me, what importance is this topic? I tried to tell them Then at the end, during the external examination, when the external examiner came in, he asked them, he said, what is the problem with this sample collection? They told him the problem we faced was that anytime we go for sample collection, the people in the offices don't allow us. It's like we are disturbing them because they have to go week after week to get the samples. Then he told them, have you taken this result back to them? They said no. He said that is why they will not allow you, because you have done the research now. You didn't give them a feedback. When somebody else wants to go, they will not give the person a feedback also. So now let's look at quantitative research. Quantitative research is a systematic investigation involving numerical data to explain phenomena. I've moved from that slide, please. So yes, so it's a systematic investigation involving numerical data to explain phenomena. And here are the characteristics. Number one, it is structured and objectives. Then it is based on measurable variables. Measurable variables, something that can be measured. Then it uses statistical tools. All these are stated while I was looking at the differences between quantitative and qualitative. Then quantitative research supports generalization of results because the data collected are quantitative. So based on that, you can generalize. But sometimes in the case of qualitative, you may not be able to generalize because the way one person is seeing something is different from the way another person is seeing it in qualitative. But in quantitative measurements, you have measured, let's say you measured the leaf area. You measure leaf area in this experiment, in another experiment, in another sample. The leaf you have measured, they are measurable. And there are things that you can generalize on. That's what I'm saying. Then quantitative research is essential in scientific studies. I'm on the next slide now. Now, what are the types of quantitative data? The number one type is discrete data. Discrete data are countable values, e.g., number of students in a class. That is discrete data. It can also be number of cattle in a ranch, number of cattle in a particular settlement, number of goats in a particular place. So if you know the number of goats, it can determine other parameters. Let's say the government is planning to have a ranch for all goats in a particular state. So when you get the number, it will help the government to be able to decide on how they will prepare for that number of cows or that number of goats. Let's say the number here is 50. In another state, it is 500. Preparation for 50 will be different from preparation for 500. Then quantitative data also is continuous data. It is measurable values with decimals, like height and weight, and other such parameters. Then we also have primary data. Primary data is data that you collected directly by yourself as a researcher. Take, for example, surveys. You went to the people or to the field to make your survey. Let's say any kind of measurement of a particular scientific experiment. You did it yourself. That is primary data. Then secondary data are collected by others, maybe by government. All right.
Can you hear me?
I can hear you.
Yeah, you are breaking at some point.
All right, okay. Secondary data, as I said, is collected by others, e.g., government record. So for secondary data, you depend on government record, which have already been collected, and use it for the purposes that you want to use it for. Secondary data also can be said to be when you look at literature that work have been done by others, and then you use that to interpret other things. That also is secondary data, and that is another type of data. Now, I'm moving to the next slide, which talk about the quantitative data collection methods. Number one way is through surveys or questionnaire. Number two is through experiments. Number three is through structured observation. Then number four is existing data or secondary sources. Now, I'm taking them one after the other. The first one is the survey. In the survey, you can use structured formats like closed-ended questions. Closed-ended question is like yes or no question. You ask yes or no question. So You now look at how many people said yes to that question. You record it. How many people said no to that question? You record it. So let's say the yes is 90%, the no is 10%, or the yes is 80%, the no is 20%. That will help you now to know how to interpret other things that you need to do with that particular data. Then the format for structure questionnaire is also, it has standardized format for all respondents. So there's no different question for anybody. Like we have in the qualitative. It based on how you meet the individual you are asking the question in the qualitative, you can modify the question. But in the case of this quantitative format, the question are standardized. Everybody must answer similar question, and the response also must follow a similar line, whether to the right or to the left. So nobody can be in the middle. Nobody can be a bit on the right or a bit on the left. So you understand that. So that is what another characteristics of a standardized format or structured formats that we look at. Now, the question types. What kind of question do we ask in this survey or questionnaire type? The number one type of question you can ask is multiple choice question. What is your age range? 20 to 25, 15 to 20, 10 to 14, and thereabout. So you ask that question, so you, based on that, you know the age range of the individuals, and that can help you to be able to interpret and determine and decide, make a decision on what to do later. Then we also have the Likert scale, and then we have what is called strongly agree or strongly disagree. Then you can use tools like Google Forms, SurveyMonkey. All these can help you to get your values. So instead of preparing questionnaire and going to the field to meet people, you can prepare Google Forms, SurveyMonkey, and share it, especially if the people you are interacting with are educated people. If they are not educated, they are illiterate, you cannot use Google Form or SurveyMonkey. They will not understand. Even the paper one you use and take to the illiterates, you have to help them to fill it. You ask them the question and you fill it on their behalf. So we have to be careful on that. Now, coming to the aspect of experiment is another way by which it's another data collection method, experimenting. You carry out experiment maybe in the laboratory as a biologist. You go to the laboratory, you do some experiment. Then you have your control group. You have the control group which does not receive the treatment. Let's say you planted maize in a pot. Then they are saying that when you use cow dung manure, it's going to allow them to grow very well, better than when you don't use cow dung. Then you prepare some pots with cow dung in them. Prepare some pots without cow dung in them, and then after 1 week, 2 weeks, 3 weeks, you measure the height of the plant. You measure the length of the leaves in the one with cow dung and with the one without the cow dung. After the experiment, you measure the length of the cob. Is it bigger than the other one? You know, it's not just looking at it with the eye and say, this one is bigger. No, you measure it, the length of the cob and other things. Or you can even weigh the cob that produced, that's the maize cob produced at the end of the day. So that will help you to know whether truly the one that has the manure is doing better than the one that does not have the manure. That is experimenting. Then the structured observation. You use checklist based on observation. You observe. Use a preset list. Number of times a student raises his hand. Then time sampling, observations at regular interval. So like this can also be said like in 1 hour between 8 and 9 in the morning, you want to count number of cars that pass a particular place. This can be used by... Yes. Yes.
Yeah, it was really breaking.
Wow.
On this structural observation, maybe.
All right.
If you can.
I'll take that again. So I was talking about the structured observation, which is a third type of data, quantitative data collection. And I said you can use a checklist-based observation. In this case, we use a preset list. For example, number of times a student raises his hand in the classroom. Then I'm bringing us to the real-world example. Between 8 a.m. and 9 a.m., you want to see how many cars pass through a particular place. Because let's say the government is planning to expand the road, and then they want to see, is there a need to expand the road? How many cars pass through that place at rush hour and when there's no rush hour? So between 8 and 9 or during the rush hour, definitely there'll be a time schedule, a time frame. You can't just say rush hour. You have to define it, 8 and 9 or 8 to 10 or 7:30 to 8:30. You have to define the time of the day. And maybe rush hour in the afternoon, 3:30 to 4:30, 3:30 to 5 p.m. Then you schedule yourself there and count the number of cars that pass the road. When there's no rush hour also, maybe in the afternoon between 12 and 1 p.m., how many cars pass the place? When you record all this, it can help the government and whoever want to work on the road to determine what they want to do at that place. All this also applies to different areas of experiment. Then the time sampling, observation at regular intervals, maybe every 5 minutes. Maybe you observe every 5 minutes. I did an experiment while I was on Fulbright in the USA, Oklahoma State University. We measured an experiment in 2 weeks. And then in 2 weeks, we saw that everything has been consumed already in that experiment. So we designed it, let's take it daily. So we took it daily, we set another one daily. So we checked day 1, day 2. Some we recorded for day 1, some were done in day 1, some were gone in day 2, another one in day 7, another one in day 21. Then we now say the one for day 1, let us see what happened. Okay, so we set another one up, and every 2 hours, we're checking it for 24 hours. And that help us to know at what time that particular compound was consumed. So another example, as I have on the slide, is observing student behavior in a science class. Now, this plate here is showing the types of quantitative data: interview, structure observation, sampling for probability, and survey questionnaire. Now, I also spoke about existing data or secondary sources. And I said these ones, you are not the one that carried out the experiment yourself. You make use of already existing data, maybe with the government. Institutional record, like WHO have certain data that of malaria cases in Nigeria in 2010, 2011, 2012, 2013, 2014. All these things are available. NCDC, Nigerian Center for Disease Control, also have different data of different issues year by year. So you can go to the database and pull this up, and then that can support what you are talking about or what you are researching on. So this is the institutional record, like the school, attendance log, and then advantage. This saves time and it also saves resources. It saves you time and saves you resources. There are some data that you don't need to reinvent the wheel. Honestly, there's no need to reinvent the wheel. Like in my area of research, there are so many researches have been done on certain things, and they have established that this is the best to use because of this reason. In fact, it was a big issue. Different people were looking at what can be used. Now, after different discussions by different scholars, they have come. This is a done deal. What is referred to as a concept of indicator species. Then I saw somebody coming to do a research and then trying to look for something that had been trashed. Assuming the person understood that concept of indicator species, the person will not be trying to reinvent the wheel. The person will just work on what has been achieved and then pull resources and then try to establish based on what has been done before and move ahead. So while the person was making the presentation, I was telling the person, why are you doing this? Does this mean you don't understand the concept of indicator species in pollution? So if you understand that concept, that person is not supposed to do that. So what I'm driving at is that when you are looking for data, try to look at already established data, so that will now make you to know what data is not yet available, which you can now pursue. So that is that about that. Now, let's come to the next slide, which talk about designing effective quantitative instruments. In this case, number one thing you need to look at is validity. Does the instrument measure what it is supposed to measure? So what you want to use to measure, you are trying to see the load on a particular bridge. Is it women that are going to count or the cars? It's the cars you are going to count that are passing the bridge, not the humans. So the instruments you are using must measure what is supposed to measure. So another thing is reliability. The instrument should be consistent over time. Then there should be no bias. It should not have any kind of bias. It should be neutral. The instrument should be neutral. Now, let's say you are making a questionnaire and then you are telling people that, like in the example I have here, you have to avoid leading question that don't you think school uniforms are better? Don't you think eating plantain is better than eating banana? You're already pushing the person to give you a particular response, which is yes, I think so. But when you ask open-ended question, um, what what do you prefer, banana or plantain? Then the person has the liberty to choose. But when you put it that, don't you think that is leading question, which will give you, which is biased, and which will not give you the accurate meaning or the accurate value of for that experiment, then your questioning should be neutral. The instrument should be neutral. What is your opinion on school uniforms? What is your opinion? Which one do you think is the best, banana or plantain? Then the person have to decide on their own. That's neutrality. Then coming to sampling technique, which is our next slide. Probability sampling is one type. In this case, you can use random sampling or stratified sampling. It's best to use random sampling so that, let's say you are having a treatment of a plant in a pot, and then you have the same plant in triplicates. The plant in triplicate should not be on the same place. You should scatter them so that this plant of zero concentration in triplicate, they should not all be on the same side. You scatter them in the different places so that in case, apart from the concentration you put in, maybe there is some other environmental factors. So if you arrange all the 0 mil together, all the 100 mil, on the 100 concentration together, it will be assumed that some environmental factors are coming from that side where you arrange them. So if you put 100 mil at this point, 100 mil at another point, 100 mil at another point, where you scatter them, that is what I'm trying to say. That helps you to get accurate value. Then there's also non-probability sampling. In this case, we refer to this as convenience sampling or purposeful sampling. This is another type. And sampling size also depends on population size and research goal. The population size will determine your sampling size. I give you an example. I worked on hydrocarbons, oil pollution, degradation, using microorganisms to clean up oil pollution. And I worked on hexadecane while I was at Oklahoma State University. I worked on hexadecane and I went for a conference. And somebody asked the question, because based on hexadecane is an alkane, and I generalized that since my microorganism, my bacteria could do this with hexadecane, that means it can do it for all alkanes. Somebody in the audience asked, you only work on hexadecane, how can you generalize? And my supervisor was there, and we went back to the lab, we said what that person said. So we went on to work on other alkanes, we worked on hexane and cyclohexane. So that helps us now to balance. You can see that. So the population size, you can't just work on one organism and then you said you generalize for all organisms. No, you have to take a series, a number of organisms, a number of tests have to be done before you generalize. Now, let's look at the ethical considerations. Number one, which is the next slide. Number one is informed consent. Participants have to know their rights and they have to give you permission. You can't just badge on the participants and be taking, you can't just badge on the participants and start taking sample from them or start questioning them. There's a skit I watch on TikTok or in the social media. This individual has a stethoscope like a medical doctor on his neck. Then he will just go to somebody on the street, maybe the person is taking water, he will take the water and throw the water from the person away. Then he'll bring out the stethoscope and be trying to put it on the person's chest. Then the next thing is bringing injection out. At the point of bringing injection, the person will now stand up and say, from where to where now? You just came and want to give me injection. Where is that done? So there must be informed consent. If you are reaching out, you want to get data from individuals. So like I was giving the example of my students, they went around different faculties and they get air microorganisms from the offices of different offices on the campus. Some people will look at it that why are you taking the air sample? Okay, today when they give you allow you to take sample today, tomorrow you go there. Those same people they were cooperative yesterday, they'll not be cooperative the next day. So, but if you have informed them ahead, this is how it's going to go. You are going to be here for so time. Please, are you sure you are going to allow us for the 13 weeks you want to take the sample? Then they say yes. Then in that particular experiment, we did not go to lecturers' offices because a lecturer that came to office today, tomorrow or next week, he may not be in the office at that time. A lecturer can travel. So what we used was general offices. So you can see you have to look at we use general offices, HOD's office, the secretary will always be there every day, Monday to Friday. Even if no lecturer is around, the secretary or other staff in the HOD's office, departmental general offices, they'll be around. So that's where we targeted because that place will always be open, be available. So another thing is that anonymity. No names or identifying details are collected. One of my students work on waste water from some factories in Ilorin, and I told her, don't write their names. Definitely, even the factories will not allow you to take their sample on a good day. But after, you have to go through the back and take the sample because they assume that when you take the sample and you find something that is bad in the wastewater, you will report them to the government. But we are doing our research on the anonymous ground. So I told the student, don't put their names. The student went to make a presentation somewhere, and then she was mentioning the names of the companies. If people in the audience told her, why are you mentioning their names? So when you are reporting, you don't need to mention the names or identifying details that are collected, especially when the information is sensitive. You can just say, we took it from so-so area, fine. But you now mention the name of the company, the name of the people, the name of this, the name of that. That is not good enough. Then another issue in data ethical consideration is data protection. Keep data secure and keep data confidential. Every data you get from individuals, keep them secure. So the next slide, I have ethical consideration in quantitative research. You have the informed consent, privacy and confidentiality, avoiding harm, then balancing benefits and risks, transparency and reproducibility, avoiding bias, reporting negative results, have to be careful. Then respecting cultural differences. As you go on, there are different cultures. So you have to respect that. In some cultures, one thing acceptable in one culture is different. So you have to understand their culture and respect their culture so that you don't have any challenge in the process. Then avoid plagiarism and misrepresentation. Then look at the long-term impact of your report. Now, I want to ask this question. I don't know if you can put it in the chat box. This example, and I have an assignment here for you also. This one, design a short 5-question survey on students' attitudes towards homework. So we are looking at quantitative. We have looked at quantitative research. Now you want to design a short 5-question survey on students' attitudes towards homework. What kind of question are you going to ask? You want to know their attitudes towards their homework. Remember, this is quantitative. So you can ask the question. Let me give the first one, then you can put the others in the chat box. I can ask the question: how many a short question survey on student attitude towards homework. You can ask the question that the homework your teacher gives you. Select one from the following: too much, too little. You can see that the homework is too much. Every time they give us homework, it is too much or too little. So if the student is saying, if 90% of the students say too little, that means the student's attitude towards homework is a good one. That means they like doing homework. But when they say too much, that the homework is too much, you know that the attitude towards homework is not favorable. So that will now help you to determine how you are going to work on the students to make them to like their homework and to see the importance of the homework and do the homework. I hope you understand that. I want us to drop other other survey questions similar to what I've dropped now. Okay, I can see that the chat box is not available. Anyway, we have to look at that. I've given you one example already. Now, let's go to the assignment. In the assignment, you conduct the survey with 10 classmates and then record and summarize the results using a simple chart or table. So the same question I've asked above, you design the question. You know, I've asked one, then you prepare the second one, the third one, the fourth one, 5 questions. Then you conduct it with 10 of your classmates, and then you record the answer. And you see the answer is either that the homework is too much, or is too little, or it is adequate. So that will help you to now determine the student's attitude towards homework. And how do you now state it? 90% said they are comfortable with the homework based on their responses. That means the attitude, they have a positive attitude towards homework. You understand? So that value will now help you to interpret and then help you to conclude or state what this object of the survey is. So I'm moving now to the conclusion that quantitative methods help us understand the world through numbers. In quantitative, everything is about number. You want to know, you want to have the number, and this quantitative is objective. Then choosing the right method and tool is crucial. Anytime you want to use any method or want to do any quantitative data collection, look at the right method you should use, and that's very important. Then ethical and accurate data collection leads to trustworthy results. When you do it ethically and then you do it in an accurate way, your results are going to be trustworthy. So that's where I'm going to stop. I thank you for giving me the opportunity to speak to you at this time. Thank you very much.
Can you give a round of applause to our presenter, Dr. Adetitun, for this wonderful presentation? Thank you so much for such a delightful presentation on this topic. So this is our time to ask questions. If we have any question, you can raise your hand up. I see that our chat we're having issues with the chat, but I will also be checking our WhatsApp platform just in case you drop a question there.